Alagappan, and I'm going to walk you through what I did for my Europe project. I set out to develop a fluorescence in situ hybridization or fish technique to visualize unfixed Thiomargarita cells. Thiomargarita are giant sulfur oxidizing bacteria that live in marine systems. They can grow up to 750 microns, making them one of the largest bacteria that we know of. The single cells of Thiomargarita are held together by mucus sheath in a string or a chain conformation. These bacteria accumulate sulfur granules so that they can use them later under nutrient deficient environments. The geobiology lab has observed endobionts within Thiomargarita, and metagenomics has revealed that these endobionts are gamma proteobacteria similar to Thiomargarita itself. Fluorescence in situ hybridization, or FISH, is a very powerful tool that can give us a lot of information about the spatial distribution of microbes within a community, or in this case, microbes within Thiomargarita. How FISH works is that we design a probe to bind to a conserved sequence in the ribosomal RNA, and this designed probe is conjugated to a fluorophore. So when this probe binds to the conserved sequence, and the unbound probes are washed away, and we view the sample under a fluorescence microscope, we can see where the ribosomes of this organism are. The purpose of this study was to develop an apparatus to perform in-solution fixation-free FISH on Thiomargarita so that we can visualize the endobionts and get an understanding of their spatial distribution within Thiomargarita. I designed a cell strainer scaffold using CAD software, and I printed it with a 3D printer. I harvested the nylon membrane from a Sigma Aldrich cell strainer and used it to make my cell strainer, as is depicted here. The cells were picked from Namibian sediment brought back by Dr. Bailey, and the cells were incubated with the UB338 probe, which binds to all bacterial ribosomal rRNA, and GAM42A probe, which binds to all gamma proteobacterial rRNA. The Alexa 488 fluorophore fluoresces green, and the Side 3 fluorophore fluoresces yellow. The cells were hybridized overnight for around 14 hours, and they were incubated at 46 degrees Celsius. Later, they were counter-dyed with nuke blue, which binds to DNA, which is a fluorescent dye that binds to DNA for 30 minutes. So in the first rounds of doing this technique, I found that I lost a lot of the Thiomargarita cells that I went in with. I was never able to find any Thiomargarita cells after I finished the FISH procedure, and I transferred the sample from the apparatus to a petri dish to look at it under the fluorescence microscope. I took very limited amount of pictures because the FISH just did not seem to work on the Thiomargarita, and I couldn't find any Thiomargarita. I think that was the biggest problem. The first picture here is of a diatom frustule, which is basically the silica cell wall of a diatom. So you can see that the green and the yellow light up, meaning that there's, maybe some bacteria and some gamma proteobacteria. However, there is a lot of nonspecific binding to the silica cell wall, which is a little suspect. So this doesn't, because there is some nonspecific binding here, I'm not sure on how concrete it is to say that the epibionts of the diatom are successfully visualized using fish. In this picture, we have a sediment particle, or sediment particles that host bacteria, and fish did a really good job of hybridizing to these microbes' ribosomes, and we are able to clearly see bacteria and the overlap of gamma proteobacteria within those colonies. Because I kept losing my Thiomargarita cells, and I just never could find any when I looked at it under the microscope, but I was very sure that I picked Thiomargarita cells. What I did was I took like chains of Thiomargarita, which are very recognizable with like a very specific number of cells. So right here we have 1, 2, 3, 4, 5, 6, 7, 8 cells in this chain, and in this one there are 4, and this is a chain of 2. So I did the fish procedure, and then even though putting the sample under the stereo microscope allows like for photo bleaching, I did this anyways just to see if the Thiomargarita were intact after the fish procedure, and it seemed that they were. So this is the membrane in the background, and this is our chain of 4, and this is our chain of 8 and chain of 2. So the Thiomargarita are intact after the fish procedure. So the problem arises in the transferring process. The apparatus seemed to work as far as the fish procedure goes. However, it did not work for Thiomargarita itself. We can see that the bacteria in the sediment clump light up. So definitely the fish worked. However, the retrieval of Thiomargarita is very, very low. Practically, it is non-existent. What we did learn from this is that the membrane pore size is appropriate for Thiomargarita. Usually the cells are anywhere from 100 microns to 180 microns, so the 20 micron pore size is appropriate. Another thing that was noticed during these rounds of experimentation was that overnight hybridization may not be ideal because the probe solutions seem to evaporate after 14 hours. So more testing needs to be done on determining the appropriate incubation time. As for future directions, I would like to improve the current design and technique. The idea that I have is to reduce the wall height and incorporate a clamp, because what I noticed was I had a really hard time dismantling the scaffold to get the membrane out, because the pipetting, especially within the 96 well plate, is incredibly hard. So I think something like, using a clamp so that, I can easily take off the washer could be a potential future direction. I would also like to try hybridization chain reaction fish, or HCR fish, once I get the apparatus to work, because HCR fish is more sensitive and it uses smaller probes. So because it uses smaller probes, it might be able to penetrate better than some of the bigger probes like the Ube probe, because Thiomargarita has multiple vesicles and multiple layers of cell membrane. So HCR fish is definitely something that I would like to pursue in the future. Thank you so much for listening to my presentation.